What we're going to be talking about today is brand new. Okay? And if you didn't notice already, I've already given you everything filled in. I want you guys to really just kind of focus on me instead of having to write and listen. This is a pretty complicated lecture. Okay? So I don't necessarily like to get the notes filled in because I know it's harder to stay attentive. So just, try your best. Okay? What we can basically describe cellular metabolism and heat balance as is a relationship between breathing and eating, and therefore the ability to make energy. Okay. Our first actual slide here is actually something we've seen before. This is a direct copy and paste of the slide we saw earlier in the semester when we first introduced to you guys metabolism, all the way back when we were talking chemistry, biochemistry stuff in Unit 1. We learned previously that the term metabolism is used to describe all chemical reactions occurring in our body cells, and that there are two principal forms, catabolism and anabolism. Catabolism is those chemical reactions breaking down complex organic molecules into simpler ones. It involves exergonic release of energy. Okay. So I always think exer begins with EX, just like exit, and release and exit kind of can be thought of as the same thing. We're releasing energy, which is useful to us. So what we're going to spend the majority of our lecture talking about, the catabolic reactions of glucose breakdown, okay, and therefore releasing ATP from it. We also learned previously that we can use the word hydrolysis, and we talked a lot about hydrolysis actually in that last lecture on the breakdown of food, okay, from like carbs into monosaccharides or proteins into amino acids. Remember, hydrolysis is where we can take something like AB yielding A plus B, and water is used to split the bonds and making two smaller or several smaller pieces. This is what allows for the release of ATP to occur. Okay. Anabolism is where we're building stuff up from simple molecules and monomers to form the body's more complex structural and functional components, which are not exergonic, but they're endergonic, meaning that they consume more energy than they produce. And this, as we know, requires more energy, or requires energy in the form of ATP. Okay. That ATP, which was formed back in catabolic reactions. Okay. This ATP that we're going to be talking about today can be formed three different ways. Substrate level phosphorylation, where you're forming the ATP directly from the substrate. Okay. This happens in glycolysis in the Krebs cycle. Oxidative phosphorylation, guess what needs to be used or present for oxidative phosphorylation? Oxygen. So that's what we're going to talk about with the electron transport chain. We won't get to that today, but we'll finish that and talk about that on Tuesday. What about photophosphorylation? We can't do this, but some, not animals, but some organisms can. Plants that can go through what we call photosynthesis. And this is going to involve dehydration synthesis, just shown down here at the bottom, which is the opposite of hydrolysis, where we remove water to go from like A plus B to AB, as we learned earlier in the semester. We just mentioned that anabolic reactions require energy, and a lot of that energy is a direct result of catabolic reactions. And this is something we talked about earlier in the semester, that both anabolism and catabolism are coupled to one another. And we can see that with the kind of cycle in our picture at the bottom. When complex molecules are broken down by catabolic reactions, some of the energy from their many bonds is transferred to form ATP. The rest is lost as heat. Now we say lost as heat, but heat is actually something that we need. It provides us with a body temperature since we are warm-blooded. And so we don't necessarily kind of think of it all the time as lost heat. So that's why at the end of the lecture we're going to be talking about thermoregulation or heat balance. Once we've broken down those simple molecules, which is like what we just did with digestion, we can take things like amino acids into our body and use ATP to build big stuff up, like the proteins in our muscle, for example. Okay. And that also will result in heat being given off. Actually, a significant amount, about 60%. So we've been talking a lot about energy transfer, and there are two important aspects of energy transfer, and those are oxidation and reduction reactions, which aid in the production of ATP. When a molecule loses electrons, we call it oxidation. Remember, electrons have a negative charge, and we can think of electrons like, well, I mean, it's potential energy. So when a molecule loses electrons, it will lose or decrease its potential energy. Now, I have kind of an analogy for electrons, hydrogen atoms, and these NAD plus FAD. I like to think of them as big brother, little brother, and then going for a car ride. Okay. I grew up, I was a little brother to a big brother. I was also a big brother to a little sister. Okay. But many of you guys probably in the same boat. Either we're a little brother or big brother, one of both. One thing, growing up, I always wanted to do what my big brother did. Okay, and so if big brother went somewhere, I always tried to tag along. Okay, and that included getting rides to school. Okay. So in my analogy here, electrons are like big brother. Where they go, little brother goes. And the little brother in my analogy here are hydrogen atoms, which a lot of times will be lost as we also lose electrons. Okay. Those electrons in hydrogen atoms will a lot of times get carried by what we call coenzymes. NAD plus and FAD, which are a derivative of some B vitamins, so coming in through our diets. We can transfer electrons into hydrogen atoms from NAD or from something to NAD plus, forming what's called NADH plus H plus. We also will sometimes call that NADH2, kind of as a shorthand. FAD can add two hydrogen atoms to become FADH2. It doesn't state it here, but electrons are also being transferred to these guys. These can basically be thought of as electron and hydrogen atom carriers. So this is like Big Brother's car. And both Big Brother, the electrons, and Little Brother, the hydrogen atoms, are going to take a ride from one place to another. Now, just like when you take a ride from one place to another, like when I got a ride with my big brother to school, when you get to school, you get out, right? And so, certain molecules will accept electrons and those hydrogen atoms, and that process is called reduction. Okay. Where you can think of reduction like a reduced charge. Because remember, if you're gaining electrons, you're gaining their negative charges. Okay, and that's where the word reduction comes from. And this causes an increase in the potential energy of a molecule. Okay. When we look at NADH plus H plus and FADH2, those are actually the reduced form of NAD plus and FAD. Okay. Because they've gained electrons. So when little brother and big brother are in the car, it's technically a reduced car. Okay. And then they dump them off, and so that makes them become oxidized. Okay. So what we do in this chemical reaction at the bottom, which happens to be cellular respiration, is we oxidize this. What is this? Anybody recognize it? C6H12O6 is the chemical formula for glucose. What we're going to talk a lot about. We basically strip electrons and a whole bunch of hydrogen, as you see there's 12 of them, from glucose. That leaves us with this, carbon dioxide, which we just got done talking about. We have to exhale out in order to maintain pH, right? Well, that's where the CO2 comes from that we've been talking about. Now, for this process to occur, we also need oxygen. Those electrons and hydrogens that were swiped from glucose end up going to oxygen, and that forms a little bit of water, what we call metabolic water. But that's not the purpose of this reaction. The whole purpose is over here on the right, production of ATP. A little bit more on oxidation and reduction. We got a figure at the bottom, or a couple figures to demonstrate what happens to those coenzymes. So when NAD plus is reduced to NADH plus H plus, or NADH2, it gains what's called a hydride ion, hydrogen with a negative charge. This neutralizes its charge, and that H plus is released into the surrounding solution. Okay. So looking here, here's our coenzyme. Here's its reduced form. We call it NADH plus H plus. It's adding electrons, but also hydrogen ions. One is directly connected. The other one is like right there, but it's not. Okay. So when we actually write it as NADH2, it's actually not technically correct. Okay. So this would be like if Big Brother was such a butthead that he made you write in the back, or in even the trunk. Okay. Or even worse, on top of the hood. Okay. You're there, but you're not there. Okay. Thankfully, my Big Brother wasn't that big. Okay. Now, when we oxidize NADH to form NAD+, the loss of the hydrogen ion results in one less hydrogen atom and an additional positive charge. So that's where the plus comes from. FAD becomes FADH2 when it is reduced. Okay, it gains a hydrogen ion and a hydride ion. It's then oxidized to FAD when it loses those same two ions. And we're going to see these reactions occurring. We're going to start with adding electrons and hydrogen atoms to them so they become reduced coenzymes, as we like to call them. Oxidation and reduction reactions are always coupled, meaning each time one substance is oxidized, another is simultaneously reduced. For that reason, we call those reactions that are paired oxidation reduction or redox reactions. Oxidation is usually an exergonic or energy releasing reaction. When a substance is oxidized, it is transferred immediately by coenzymes to another compound. This is how NADH plus and FAD help out. They carry the electrons and hydrogen atoms to the electron transport chain, which is where the bulk of our ATP is produced. Here we can see NADH plus H plus and FADH2. That's their reduced form. They're becoming oxidized because they're donating electrons and hydrogen ions to form NAD plus and FAD. Okay. And that way they can go back like a car and pick up Big Brother and Little Brother again. Make sense? Somewhat. I know it's going to be kind of murky, okay, but once you kind of sit down with it, it's tough. Okay, the majority of this lecture is going to be all about carbs. We will talk about fats and proteins because we know those are also fuel nutrients. But our body prefers carbohydrates. Okay. And so actually a little bit of this is a review from just what we got done talking about in that last lecture. So before glucose and other monosaccharides can be used in the body, they must be absorbed from our small intestine by a process called sodium cotransport, where sodium and glucose get pulled in together at the same time. Okay. And sodium acts as a way to piggyback ride or give glucose a piggyback ride. Okay. Once in the circulation, so after they've been pulled into the cell, they'll make their way into the blood. The fate of glucose depends on the needs of our body cells. A lot of that glucose is going to go directly into the cells for ATP production. And this is what we're going to talk about. But in certain states, we can use glucose to synthesize amino acids, to synthesize glycogen, a process called glycogenesis, or even a process called lipogenesis, which is the formation of triglycerides. Hopefully by now you guys know what those terms mean, because we've looked at them several times this semester. Remember back when we talked about the endocrine system, I wrote up 6 words and gave you definitions. I'm going to go ahead and do that again for you guys. They're all going to either end in lysis or genesis. We're going to talk a lot today about glycolysis, which is the breakdown of glucose to form ATP. That would be an example of catabolism. This is a breakdown. We can also perform anabolic reactions related to carbohydrates. Okay? And that's what glycogenesis is. First, I want to define glycogenolysis, which is glycogen breakdown into glucose molecules Neither of those two definitions are listed on this page, but we will get to both of those processes. We do have listed on this page glycogenesis, which is where we form glycogen from glucose molecules. Our fourth definition is something that we've seen before and we'll see again, and that is gluconeogenesis, which is glucose formation from non-carb sources. Things like amino acids and fats. And then that leaves us with our last two definitions. Lipogenesis, which is listed here, the formation of fats. Fats, we're talking here triglycerides. And lipolysis, which is breakdown of fats. And we're talking triglycerides again. So, those 6 terms will get used throughout this lecture. So hopefully by now you already know them. In case you forgot them, I went ahead and wrote them up on the board. Here we can see 2 out of those 6. Formation of glycogen, what we call glycogenesis. That's something that we do when we are ingesting a bunch of carbs. Like carbo loading before a big race, you're going to promote glycogenesis. Okay. If you eat too many sugars, what you're going to end up doing is getting fat. Okay, because you're going to promote too much of this. We can actually convert carbs into fats. Okay, that's lipogenesis. Over on the right, we show how glucose gets into our body cells. That way they can then be used, or that can be then used to make ATP. Okay. Glucose entry into most of our body cells occurs by way of what we call GLUT transporters. Okay. GLU, capital T, is short for just glucose transporters. Okay. There are several different types, GLUT1s and 2s and yada yada yadas, but they bring glucose into the cells by way of facilitated fusion. It's a carrier or transporter protein, and it's promoted when we release insulin. When do we release insulin? When we are hyperglycemic, which meant what? High blood sugar. Good. Because this is a great way to lower blood sugar, take it into your cells. Now, once in our cells, the glucose will get stuck because we end up adding a phosphate to it by way of a kinase enzyme. And those same transporters will not carry a phosphorylated glucose. And so that's when they get stuck. And then that's going to provide us the opportunity to then perform that cellular respiration. Cellular respiration is the process releasing energy from food to produce ATP. In other words, it is known as oxidation of glucose to produce ATP. Oxidation, which meant what again? A loss or a gain of electrons. Loss of electrons. Okay. It is that chemical formula you saw a little bit ago. This time we added heat at the end of it because heat is one of the energy products. And that's why we talk about heat in this lecture and heat balance. Okay. C6H12O6 is the chemical formula for glucose. Now this is a simplified version. To be technically correct, we'd say 6O2, which is the oxygen that we breathe, plus 6H2O plus 6CO2 yields ATP. And that's the whole goal. The whole purpose of the reaction is to produce ATP. Glucose contains potential energy, and when it is broken down to carbon dioxide and water, this energy is released in the form of ATP and heat. Now, where does that oxygen come from? Breathing. Where does glucose come from? Food. What two lectures did we just get done talking about? Respiration and digestion, right? Hopefully now you see why we are going in the manner that we are. And we also talked about carbon dioxide, how we have to get rid of that. Well, that carbon dioxide is produced during this chemical reaction. And therefore, it must be circulated to our lungs in order to be exhaled. That water that is produced, very little amounts, is called metabolic water. Helps actually meet some of our daily need for water, but are we going to survive on that alone? The answer is absolutely not. Obviously, we still have to ingest water, right? Whether you're drinking it directly or some other fluid that contains it. In actuality, a lot of our food is very, very watery, even though it might be really dry. Energy in the form of heat gives us a body temperature. We'll talk more about that at the end of the lecture. And then that ATP that is formed can be used for anything that requires energy. And we've talked about several things this semester that require energy. Okay. We just talked about active transport, where you pump something against its concentration gradient. That's a transport process that requires energy. Mechanical forms of movement require energy. Remember back when we talked about formation and breaking of cross bridges, what was needed to cock that myosin head and then to break it free from actin? ATP. Okay. And then ATP, we also just talked a little bit about just a second ago, was its role in forming larger things in anabolic reactions. We said that catabolic reactions help form ATP, which can then drive anabolic reactions. So a lot of different things can be formed or done thanks to the ATP we produce. ATP, as we know, is a adenosine plus 3 phosphates formed when you take a phosphate and add it to ADP, which is adenosine diphosphate. Breakdown of glucose summarized above is not quite that simple. However, it involves a complex series of equations where glucose is taken apart piece by piece, removing hydrogens and splitting the carbon-carbon bonds, thereby eventually releasing energy of glucose gradually, so that at least a significant portion, around 40%, can be made into ATP. Here on the next page, we can see a figure and then 4 steps listed on the left, which are the 4 steps or 4 sets of reactions involved with cell respiration. One of those words over there on the right that I wrote on the board is listed here on step 1, glycolysis. Set of reactions actually takes place outside of our mitochondria, which you can see here is a mitochondrion. We remember back at the beginning of the semester learned that mitochondrion is where we form ATP. It's the powerhouse of our cell. We can actually form a little bit of ATP from glucose through the process of glycolysis outside of the mitochondria back out in our cytosol. But we don't form very much, only a couple. What we do form are some of those reduced coenzymes as we steal some electrons and hydrogen ions from glucose. We can then take the product that's formed during glycolysis, what we call pyruvic acid, and form what's called acetyl-CoA. Step 2 is sometimes described as the transition reaction, because it's between glycolysis and the next big reaction, which is step 3, the Krebs cycle. The Krebs cycle is a cycle, obviously based on its name, where we produce a little bit of ATP, even though it's not really, actually, yeah, it is shown right there. A little bit of CO2 is produced here, but we form a lot of those coenzymes. Now, you notice those coenzymes that are formed in those first 3 steps are all headed by way of arrows to step 4, what we call the electron transport chain, which is where we actually take the oxygen that we breathe in and by way of oxidative phosphorylation produce ATP. This is where the bulk of the ATP that we produce is actually produced. Okay. And this is where the water that we talked about that is formed is also produced in very small amounts. So what we're going to do now is go through those processes step by step and learn more about them. We are obviously not going to get to them all today since we've only got about 5 minutes. But our first step is glycolysis, where we take a molecule of glucose. Anybody remember how many carbs were in it? How many carbs are in a singular glucose? C6, page 1206. 6 carbs, and we split them down into 2 molecules of pyruvate or pyruvic acid. You'll see those kind of terms interchangeably, which both are 3 carbons in length. So we basically split glucose in half. Okay. Here on the next page we can show exactly what occurs. Okay. This is a figure actually from the biology textbook. I like it a lot because it's really simple, but it shows what happens. There's basically 2 phases to glycolysis. One where we take those 6 carbons, we use some energy, and we split them in half. If you remember back when we talked about phosphates, we learned that they had a lot of negativity to them because they're negatively charged. And what do we know about like charges? Are they attracted or do they repel? Like charges repel. So when we stick a phosphate on both ends of a glucose, it's like bending a stick. And if you bend it strong enough, what's going to happen? It's going to snap. Okay. So what ends up happening is we form 2 3-carbon molecules with a phosphate. We add another phosphate. Those 4 phosphates will end up getting donated to ADPs to form 4 total ATP. But why do we say in parentheses here a net of 2 ATP is formed? I know most of you guys, probably all of you guys, are too young to go to the casino. Okay, but everybody in here, I'm sure, is aware of how a slot machine works. You put some coins in, you pull a lever, hopefully some more coins pop out. Okay. I talk a lot about my wife in here. I love her to death. When we go to the casino, every once in a while, not very often, she loves to play the slot machines. Okay. Her theory on how things work, she puts 2 quarters in, pulls the lever, and 4 quarters comes out. She thinks she won $1. How much did she actually win? 50 cents, right? Because you started with 2, and now you have 4. You're only up 2. We had to put 2 in, so we're already down 2. We come out with 4 coins, so that's only a net of 2. Not very much energy can be provided this way. What the main thing is we're going to steal some electrons and some hydrogens to form these 2 NADH2s. And those are going to eventually get utilized in the electron transport chain. Now, this is an example of substrate level phosphorylation. It is not occurring in the mitochondria because it happens in the cytoplasm, and it is an anaerobic process, which, as we know, means requires no oxygen. What we will pick up talking about is what happens after we formed pyruvic acid. The fate of pyruvic acid will be illustrated here first thing on Monday. If you really want to get crazy with it, these are the actual 10 steps of glycolysis, each of which requires different enzymes. The book actually does get into it in pretty good detail. Fortunately for you guys, I'm not as mean as the textbook. When I took cellular and molecular biology, I had to learn all those 10 steps, but I'm not going to worry about you guys having to do it. We'll pick up and talk about the fate of pyruvic acid first thing on Monday.